In Lecture 6, we will be looking at cellular reproduction. Okay, we're going to look at a process called mitosis, which is basically how a cell splits into two. But before a cell can split into two, it must copy all of its genetic material, right? And so we will also be looking at the process of DNA replication. But first, a little bit of history. Okay? In the mid-1900s, although it was clear that the nucleus contains the hereditary material, but the exact chemical nature of the hereditary material was not known. Scientists kind of suspected that DNA was indeed the hereditary material, but they were uncertain. The nucleus contains the chromosome, which is known to be responsible for inheritance. However, as you remember from earlier lecture, the chromosomes are actually consist of both DNA and proteins. Chromosome is just very compact DNA, and they're able to make themselves compact because they wrap themselves around the protein. So is it the protein or the DNA that's responsible for inheritance? So there was a famous experiment by Alfred Hershey and Martha Chase that was done in the 1952 that kind of helped put this debate to an end. In their experiment, Hershey and Chase used a type of virus that would infect bacteria. They are called bacteriophages, or just phage for short. And the phages would have a protein coating and a DNA core on the inside. As you remember, DNA is going to have a carbon, hydrogen, nitrogen, oxygen, and phosphorus in it, whereas the proteins, they have the carbon, hydrogen, nitrogen, and there is a little bit of sulfur in the protein as well because of one of the amino acids containing sulfur in them. So what happens is the Hershey and Chase, they grew the bacteria, the bacteriophage rather, in the different radioactive isotopes. Okay, so one type of phage were grown in radioactive sulfur called sulfur 35. So radioactive just means that you can detect them. Okay, because they kind of give off radiation constantly. And then the other type of phage were grown in radioactive phosphorus. So phosphorus 32. Which means for the first type of phage, the proteins would be radioactive, and on the second type, the DNA would be radioactive. So how does the phage work? The phage basically will attach to a bacterium, and then it will inject something into the bacteria that will allow it to replicate itself. So Hershey and Chase were trying to determine what exactly is it that they're injecting into the bacteria. So in the first group, they were labeled with sulfur 35, which means that the protein coating is going to be radioactive. So they allow these phages to infect the bacteria, and then they would actually put this in a blender. Okay, it's kind of like a low-tech lab back then, I guess. And the blender would cause the virus to detach from the bacteria. Okay, and then they would spin it down in a tube, and so the lighter virus will be on top. And then the heavier bacteria would be at the bottom. Okay, so because the virus were allowed to infect the bacteria, these bacteria would now contain whatever the virus has injected into them. Okay, and then they would try to detect the radiation. Okay, to see where the radiation is coming from. So in the case of sulfur 35 labeled bacteriophage, they were able to detect the radiation in the virus component. Okay, so that suggests that to them that the protein part of the virus actually remained outside. They didn't go into the bacteria. But just to be sure, they then repeat the experiment with the phage that has been labeled with phosphorus 32. So these are radioactive DNA molecules that is labeled in the virus. So again, they let the virus infect the bacteria, and then they use the blender to separate them, and they would put that into a tube and separate the virus from the bacteria. Okay, so this is the virus at the top because they're lighter, and then this would be the bacteria, and they try to detect the radiation, and this time they only detect the radiation in the bacteria, which means the thing that actually went into the bacteria was the DNA. And so the results from this experiment and this experiment confirms that the protein is the part that remained out of the bacteria, and it is the DNA that went inside that causes the bacteria to start producing these viral parts. And so that the conclusion from this experiment is that DNA and not protein is the material for hereditary. And you can see these are actually the bacteriophages, and that's the DNA that is being injected into the bacteria. The next big thing that needed to be done was to determine the structure of DNA, and that was based on the work of Rosalind Franklin as well as Watson and Crick. So Rosalind Franklin, she used X-ray to study the molecular structure of DNA, and her data actually suggests that the DNA is a double helix. But unfortunately, she wasn't able to finish her work. She actually died of cancer, probably because of X-ray exposure from her work. And so her data was used by Watson and Crick to build the double helix of DNA model. And they actually won the Nobel Prize as a result of that. So let's recap some of the properties of DNA before we talked about DNA replication. There is something called Chargaffs Rule. We didn't call it by this name when we learned it earlier in the course. I just kind of told you that there is a base pairing rule, and that A always pairs with T and G always pairs with C. But that was actually created by this guy, the Erwin Chargaff. Okay, so he figured out that in any given cell, the proportion of adenine always equals to that of thymine. So the percentage of A always equals to T, and the proportion of guanine always equals to that of cytosine. And so G equals to C. And then he came up with this equation, A plus G, the amount of adenine and guanine together is always going to equal to the amount of thymine plus the amount of cytosine. And that means the DNA molecule is actually complementary. Okay, so if you know one side of the strand, you can use the Chargaffs rule, you can use the base pairing rule to figure out what bases are going to be found on the other strands. Okay, so you can see the nucleotides, the base of the nucleotides are actually going to be held together by these hydrogen bonds. Okay, so hydrogen bonds are basically interaction between two separate molecules where one of the molecule is going to be slightly positive and then the other molecule is going to be slightly negative. Okay, so same thing over here, slightly positive, slightly negative. Okay, so there are two hydrogen bonds between A and T. Do try to remember that because it's going to be important for our discussion later in another lecture. Whereas guanine and cytosine, there are going to be three hydrogen bonds holding them together. Okay, so this is the slightly negative, slightly positive, slightly negative, slightly positive, slightly negative, slightly positive. Okay, and so that is the complementary base pairing. Okay, that if you know one side is A, the other side is always T, vice versa. And if one side is G, the other side is always going to be C, and vice versa. There is also this concept of antiparallel. Okay, two DNA strands are going to be parallel to each other, but one is going to be running in the opposite direction compared to the other. And this arrangement is actually necessary in order for us to maintain a constant distance between the two strands. If the DNA molecule is not spaced equally along its entire length, then that's going to create instability and it might unravel easily. So we don't want that. Okay, so to keep them constant, we require this so-called anti-parallel arrangement. So one strand is going to be running from 5' to 3'. The 5' end is called 5' because the phosphate group in the nucleotide is attached to carbon number 5 in the deoxyribose sugar. Okay, and the other end is going to have a hydroxy group, the OH group, attached to carbon number 3. Okay, so you don't really have to know why they're called 5' and 3'. again, it's just because of the carbon number that it's attached to. What's important is you know that one strand it's going to run from 5' to 3', and then the other strand is going to be running from 3' to 5'. Okay, so kind of head to toe from on one strand, and then the other strand is kind of toe to head. It's opposite. So DNA molecules are both complementary as well as anti-parallel. So here we're going to talk about DNA replication, which is something that needs to happen before cell division. The model of DNA replication was actually discovered by these two scientists, Messelson and Stahl. We are going to be looking at their experiments as well. So here we have the parent cell. The parent cell is the cell that will be dividing into the daughter cells. And in this parent cell, there are 2 chromosomes. And so what happens is through DNA replication, the 2 chromosomes are going to be copied. And so there will be 4 chromosomes. And cell division or mitosis will then cause this cell to split into 2. And each of the daughter cells will get just 2 copies of DNA, which would be exact copies of the original found in the parent cell. Okay, so we must copy the DNA in the exact way because the DNA is the blueprint of life, right? It's what makes the cell, gives the cell its identity and its function. So we want the replication process to be very exact. So there are 3 proposed model of DNA replication. The semi-conservative model, where the parent strand is going to separate into 2 and each is going to be used as a template to create new DNA. And you can see after one division, half of it would be consist of old material shown here in pink, and the other half will be consisting of new material shown here in blue. And in the conservative model, you would have the parent DNA, but rather than splitting into 2, it's just going to be cloned completely. So after the first division, you will have one DNA that is composed of entirely original material, and then the other one is going to be completely new material. So as a loose analogy, this would be like you trying to photocopy a textbook, and after you photocopy let's say a page of notes. Okay, you copy one page of notes, and after you photocopy it, you rip the note in half, right? and you stick the old half with the new half. Okay, the original with the one that you photocopy, and then you create a one whole piece. Okay, so like you have this original note, and then you're gonna make a copy, and then afterwards what happens is you will cut them in half, so your copy will be half original and half new photocopy. Okay, and then over here same thing. Okay, in the conservative model, it would be like original and then copied, and then you just keep it the way it is. So you have the original and then you have the copied. Okay, and in the last model, you have what's called dispersive model, where instead of having one strand that's entirely old and one strand that's entirely new, it's kind of dispersed all over the DNA molecule. So it's kind of like a mosaic. So if you have original and you have the copy, what this would look like is, you kind of shred them into pieces and you randomly patch them together with new material and old material. So kind of like this. Right? So which one is the correct one? Well, that's going to be determined by the Messelson and Stahl experiment. So in their experiment, they cultured E. coli bacteria for several generations in a medium containing heavy nitrogen. Okay, so normal nitrogen has a molecular weight of 14, but these heavy nitrogen has a molecular weight of 15. Right, so nitrogen 15. So after several generations of growing there, all the DNA is now going to have heavy nitrogen incorporated into them. Okay? And then they transfer the E. coli into a culture that contains nitrogen 14. Okay? And then they let them grow there for 2 generations. And after each division, they check their DNA to see how heavy it is. So I'm going to try to explain this to you on on a note over here. Okay, so again, feel free to draw together with me. Okay, so just a couple of things first. If you grow the E. coli in a heavy medium, okay, I'm going to use red to represent the heavy nitrogen 15. Okay, then its DNA is going to be heavy. Okay, so this is nitrogen 15, and so if you try to collect the DNA in what's called a density gradient, which basically it's a type of medium that will allow you to separate the DNA based on how heavy it is. Okay, so if you were to put this into the gradient and spin it down, you would detect the nitrogen 15 at the bottom. Okay, because it's heavy, right? Okay, and on the other hand, if you have the nitrogen 14, okay, if the bacteria is grow exclusively in nitrogen 14, and you put it into the gradient, then you would expect to find it at the top because 14 is lighter than 15. Okay, so light stuff goes on top and heavy stuff sinks to the bottom. Okay, now if you have a DNA that's kind of hybrid, okay, half is made up of nitrogen 15 and then the other half is made up of nitrogen 14. Okay, I don't even know how to draw the middle part. I guess we'll do part red and part blue like that. Then you're gonna have a mixed band that's gonna be in the middle. Okay, so this one is going to be not as heavy as 15 but not as light as 14 because it's half 15 and half 14. Okay, because think of it as kind of like 14.5. okay, so it's going to be hovering in between. So what they did is they want to use this method to figure out whether the DNA replication model is semi-conservative, conservative, or whether it is dispersive. So this was from the slide earlier, and the pink is where the N14 medium was used, and the blue represent new materials, right? So after the first division, after the first replicative cycle, if you were to check the DNA in those gradient tube, what results would you expect? Well, if it's a semi-conservative model, you would expect to have a mixed band in the middle, right? In the middle. Okay, in the middle right there. Okay, because it's half-half. If it was the conservative model, then after the first cycle and you run the DNA in the column, you would expect to find a light band at the top because of this DNA, and you would expect to find a heavy band at the bottom because of the original. If it's a dispersive model, you would also expect to find something in the middle, okay? Because the DNA is half new, half old, but the new and the old are kind of mixed up in both strands, okay? As opposed to definitively one old strand and one new strand. Okay, this is kind of dispersed all over the place. That's why it's called dispersive model, right? And so they did that. They did the experiment, and what they saw was a mixed band in the middle. Okay, so that tells them that the conservative model is not correct. Okay, because if this were correct, they would expect to find two bands, right? But they didn't. After first replication cycle, they only found a single mixed band in the middle. But that doesn't help them distinguish whether it is semi-conservative model or dispersive model. And so they had to do another cycle. They have to wait another replication cycle. So in here, in the semi-conservative model, you can see the pink and the blue will separate, and the pink strand will now build another blue strand using nitrogen 14. This blue strand was made up of nitrogen 14, and since the E. coli are only growing in nitrogen 14 now, the new strand will also be composed of nitrogen 14. And so if this was a correct model and you run it on a column, you would expect to find a light band at the top, right, because of this strand, and you would expect to find a medium band in the middle. Okay, and over here, so we're not going to talk about conservative anymore, right? Because we know it's not true, because we rejected it in the first cycle. For the dispersive model, what would you expect? Well, in the dispersive model, you can see the DNA is still kind of half some pink and some blue, and it's not distinctively one strand pink, one strand blue. That means all the DNA are going to be composed of both heavy and light nitrogen, and so you will expect to find a mixed band in the middle. Okay, and so when they did the experiment, they found that there were two bands, okay, a light band and a mixed band in the middle, as opposed to just one mixed band. So that means the dispersive model was not correct, and the semi-conservative model is the correct model for DNA replication. So the conclusion, DNA replication uses semi-conservative model where each of the two daughter molecules will have one old strand derived from the parent molecule, the original DNA that is, and one newly made strands, and the two strands are going to be connected lengthwise. And since two strands of DNA are complementary, which means each strand can act as a template for building a new strand in replication. Now that we know the DNA replication process is semi-conservative, we are going to look at the mechanism of DNA replication. That is exactly how you copy the DNA and create two copies of it. So here in this picture, this is actually a whole strand of DNA right there, okay? And what happens is there is kind of like a bubble that opens up in the middle, and this is where DNA replication are going to be starting at. Okay, so these bubbles, they're called replication bubbles. So in DNA replication, the parent molecule is going to unwind, and the two new daughter strands are going to be built based on base pairing rules. Okay? So again, A goes with T, G goes with C. And the copying of DNA is actually very remarkable in its speed. It's able to copy 1,000 base pairs per second. Okay, think about typing, right? Like how many letters can you type per second, right? Compared to your DNA replication speed, 1,000 base per second. And it's not going to be making it's not going to be making mistakes either. I mean, sometimes it does make mistakes, but for the most part, it doesn't. And so its accuracy, it's quite high. You have to copy 3 billion base pairs, right? Every time you copy the entire DNA genome in the cell, and you have to do it at such high speed without making mistake. So it's quite a remarkable process in that regard. So there are more than a dozen of enzymes and proteins that participate in the DNA replication process, and of course we're going to be looking at some of these enzymes. So here you can see the original parent DNA molecule. We are going to be making a copy of this DNA molecule, and to do that we are going to have to open up some replication bubble. Okay, so you are gonna pop open here, here, and here. Right? It's kind of like, you know, sometimes I don't know if your zipper of your jacket ever pop up right in the middle, and then you can kind of stick your finger in and run it along the entire length of the zipper to unzip it. This is kind of what's going on. So by opening these bubbles, we are going to be able to add new nucleotides to it. So these blue lines, they represent the new strands that we are building. Okay, and so the replication bubbles, they're going to expand laterally, and eventually this blue strand is going to join with this blue strand. Okay, so you can see the bubbles getting bigger and bigger, and once they join together, then you will have two strands of daughter DNA. Okay, each strand consists of old material, shown here in purple, and one strand of new material, shown here in blue. Okay, so that's basically the overall process of DNA replication. This is basically what I just described. Go ahead and read that on your own. Here is a list of key enzymes that are responsible for the DNA replication process. We'll be looking at each one of them in more details coming up. So first, we want to open up the DNA, right? We want to create that replication bubble. So we're going to have to unwind the DNA. And to unwind the DNA, we need to have this enzyme called helicase. Okay, so helicase is the enzyme that will unwind the DNA at the replication bubble. However, as you unwind the DNA, as the bubble continues to expand laterally, that's actually going to create quite a bit of tension in the DNA. It's kind of like a rubber band, right? If you keep on twisting it, then it's going to create all these coils and tensions. So we don't want the DNA to coil up and tangle up and have tension in it. And so we have another enzyme called gyrase, which will be relieving any tension that builds up from the unwinding. Also, the DNA strands, because the bases are going to be attracted to each other, since the A are attracted to the T and the C's are attracted to the G's, and so if you separate the DNA molecules, they actually have a tendency to kind of close up and come back together to form the double helix. So we don't want that to happen. That's why we will use these single-stranded binding proteins to keep the strands apart. So here you can see this is one side of the replication bubble, and we actually call this the replication fork. And so you have the number 2 here. That is actually the helicase, which is responsible for unwinding the DNA, and downstream from the bubble, you have the number 1 here. That's the gyrase that will be relieving the tension as the helicase unzips more of the DNA. Okay, and to keep the 2 strands apart from each other so that they don't recombine, we have these circle proteins. They are called single strand binding proteins. Okay, so unwind the DNA helicase, prevents the buildup of tension, gyrase, and to keep the 2 strands apart, we have the single strand binding proteins. The actual enzymes that's responsible for finding the right nucleotide to put on the DNA strand is called the DNase polymerase 3. Okay, so if I have A, T, C, G, T here, the other strands would be T, A, G, C, A. So now this is going to be the replication fork, and this is the bubble. We need an enzyme to find the complementary base pair, right? So something has to put a T here, A here, G, C, A, and then on the other side they have to put the A, T, C, G, T, like that. So you will be able to have 2 strands afterwards, right? So that enzyme is called the DNA polymerase 3. So DNA polymerase 3 uses the template strand. Template is the original to synthesize, to create the new daughter DNA. Okay, so this is the daughter DNA, right? This new one here, right? But the enzyme has 2 big limitations. Okay, so remember, the DNA is antiparallel. So if this is 5' to 3' here, then the bottom strands would be 3' to 5'. Okay, so it's antiparallel, which means when you build the DNA strand, this side is going to be 5' because this needs to be antiparallel to the original, and this is going to be 3'. And on the bottom here, we are going to have the opposite, because this is 5, this is going to be 3', and this is 3', which means this is going to be 5'. And the problem with DNA polymerase 3 is that it can only build DNA in the 5' to 3' direction. Okay, so in the top here, right, you are going to be able to continue to add nucleotides in this direction because you can add things on the 3' end, right? And over here you'll be able to add nucleotides in this direction, okay, on the 3' end. So that means if these bubbles were to continue to open up, then you won't be able to add nucleotides on the 5' end here. Okay, because DNA polymerase 3 can only add on the 3' end. So how are you going to replicate the rest of the DNA here, right? Similarly, how are you going to replicate the rest of the DNA here? So that's the first problem that we need to solve. The second problem is DNA polymerase 3 can only build DNA if something already started the process. Okay, if there is no nucleotides on a strand, if this is like the beginning, like this, right, and DNA polymerase 3 come, they won't be able to do anything because there's no letters that are put on there. There's no nucleotides that are put on there, so they kind of don't know where to start. Right, so something else has to start the process. So there's an enzyme called primase or RNA primase. That is the enzyme that will lay down RNA primers so that DNA polymerase 3 will know where to start. Okay, so that solves problem number 2. Okay, DNA polymerase 3 doesn't know where to start. The primase will put down a few nucleotides in the form of RNA, and that will guide the RNA polymerase 3 to know where to start. The next the next problem is that DNA polymerase 3 can only add nucleotides on the 3' prime, right? Which means one of the strands are going to be produced continuously, right? And that strand will be called leading strand, whereas the other strands are going to be produced in fragments, and that is going to be called the lagging strand. So let me try to explain this a little bit better on a diagram, perhaps. Hopefully, it will make more sense. So let's draw this together, if you could, right? So this is one side of the replication bubble. Okay, one side of the replication bubble, and I'm gonna put some letters there, so it will make more sense. So we have A, T, C, G, you know, whatever. I'm just gonna make up a bunch of letters, and you can do it with me if you want. So this is going to be 5' to 3', which means at the other end is going to be 3' to 5'. And they are going to be complementary to each other. So the A here will be having a T on the other side, right? C will be G, and so G is C, G, A, T. Okay, I'm just going to be writing down the complementary strands. So these are the parent strands. Okay, these are the parts of a DNA that haven't been separated yet, right? And you know, somewhere down the stream, you might have like a helicase, right? Helicase. That will open up the bubble more for you. Okay, and so what happens is the DNA polymerase 3 will not know where to start. It requires, you know, some kind of guidance. Right? So what happens is there's going to be the primase. Okay? The primase is going to come along and put down a few nucleotides in the form of RNA first. Okay? So the primase will come, say here, and it sees an A. So A pairs with T. But this is RNA primer, so we don't have T. Instead, we'll have uracil. So we'll put down like a U here. It will put down A here, and then it will put down maybe like a G here. Okay, so this small chunk of RNA is going to be called the RNA primer. Okay. So once you have the primer put down, and I'm going to put another primer here. This is going to be UAG. That's another primer. Now the primer also has directionality to it. Okay, so if this is 3, this is going to be the 5' end. This is going to be the 3' end. If this is 5', this is going to be the 3' end and the 5' end. Okay, so now the DNA polymerase 3 is going to come, right? It sees the primer. It will know where to start now, but it can only add on the 3' end. Okay, so what happens is it will add another one here. Okay, G. It will add another one, T, A, G, C, G, G, T, A, which is great because this looks exactly like the other strand, right? So you're copying, you're creating the new strand. And as the helicase open up the bubble a little bit more, right, you will be able to continue to grow into the fork. Okay, because this is the 3' end, right, and you can continue to add on the 3' end. So this strand that has been created continuously, this is called the leading strand. Leading strand. Okay. So on the other hand, the DNA polymerase 3 will be able to add another nucleotide here, and then it's going to be able to put T. Remember, we're doing DNA now, so there's going to be T here and A here, and then it's going to be able to make more on the other side. However, as the bubble continues to open, you won't be able to build backwards because you can't add on the 5' end. The DNA polymerase 3 does not know how to add on the 5' end. And so how do you replicate this section? Well, to replicate this section, you will need to put more primer. So another primase might have to come, and then it will have to lay down a few nucleotides for you. So, you know, put down a U here, and then A, and then a C, right? So it's going to put down a primer. And so the DNA polymerase 3 will now be able to, you know, put in some nucleotides. Okay, so you can see this is one fragment and this is another fragment, right? As opposed to being synthesized continuously, this strand is being created in chunks, right? So this is called a lagging strand. Okay, and these fragments that you are creating along the way actually has a name. Okay, they are called Okazaki fragment. Okazaki fragments. Okay, so let's just do this in another view. Okay, let me try to do this in another view for you. If I have the replication bubble, okay, this is 5 prime to 3 prime, and this is 3 prime to 5 prime. Okay, and you have the helicase here unwinding. You have the helicase unwinding. So what we saw was that we will put a little primer here, okay, shown here in red, and the primer is going to be anti-parallel to the original strand. So if this is 5, this would be 3, and if this is 3, this would be 5, and then the DNA polymerase 3 will be growing the strands continuously here, okay, because you can always keep on adding on the 3 prime strand. So this is the leading strand. On the other side, right, if this is 3 prime, then you will be putting it down a primer. So this will be 5 and 3, and so the strands will be able to grow in this direction. But as you open up more of the fork, you will need to have another primer, and then you will be able to grow here, and then you will need another primer, and then you'll be able to grow here. Okay, so this strand here is called the lagging strand, because you are producing the new strands in fragments, in these Okazaki fragments. Okay, but if you look at the other side of the fork, this segment that is at the front, that will actually be able to grow continuously, right, on this side, because now this is the 3 prime end, right, and you can keep on growing the 3 prime end. So the lagging strand on the other side of the fork would become the leading strand. Okay? And over here, as the helicase open up more, you will need to have another primer and another primer that will allow the strand to be produced, right? So the leading strand on one side is actually the lagging strand on the other side. Okay, so don't get too confused by this whole thing. The main thing to know here is the leading strand is being produced continuously, right? Whereas the lagging strand is being produced in fragments, called the Okazaki fragments. So here's the nicer diagram again. We see the primase represented by the square here, putting down an RNA primer that will allow the DNA polymerase to know where to start. And on the lagging strand, you are going to be producing these new strands in fragments, whereas on the leading strand, you're going to be able to continue to replicate into the fork. So what happens is when the Okazaki fragments is being built, eventually the DNA polymerase 3 will approach the primer that is in front of it. Okay, so at that point, what happens is the DNA polymerase 3 will fall off the strand, and it's going to be replaced by another enzyme called DNA polymerase 1. So the DNA polymerase 1 will remove the RNA primers and replace them with the appropriate nucleotides. So if we go back to our drawing here earlier, okay, the DNA polymerase 3 will continue to build C, G, and then C, but when it approaches another primer, the polymerase 3 will fall off, and then the DNA polymerase 1 will come on, and DNA polymerase 1 will remove the primer here. So it will remove the U, okay, and replace it with the appropriate nucleotide. In this case, that would be T. Okay, we'll remove the A, right? Even though it was correct, but they will still replace it anyways, and then replace the G here as well. So now this segment here replaces the original RNA primer. So that's the job of DNA polymerase 1. Okay, but still there is going to be a missing a bond here that connects this fragment with this fragment, and so there is going to be another enzyme called ligase that will come and physically connect them together. Okay, so DNA ligase joins the gaps in the Okazaki fragments. Is going to be able to form the covalent bond, the sugar phosphate bond, right, between the two nucleotides. In addition, both DNA polymerase 1 and 3 will be able to proofread the newly synthesized strands, and they are going to be able to correct any error. So, for example, if it's ATCG, and then the DNA polymerase 3 is building, so it's going to put a T, A, and then the next one, they accidentally put a T in, right? They're able to detect that mistake, right? Kind of press backspace if you would, and then put in the new one. Okay, so this kind of proofreading is important because if you don't correct the a mistake, then it's going to result in permanent changes in DNA sequences. Okay, so usually they are able to pick up these changes, but in some cases they don't, and that's how you get mutations. Okay, and mutations can have long-lasting ramifications. We're going to look at mutations in another lecture. So here you can see the Okazaki fragments here. After the DNA polymerase 1 replaces the primer, you still need to join the gaps together, and that's what the DNA ligase is going to be used for. This slide summarizes everything that we've just discussed. It's a good summary slides for you to read through yourself, so make sure you do so. There is one limit in terms of the DNA replication process. So following each DNA replication, the ends of the eukaryotic chromosomes actually will get shorter and shorter. Okay, prokaryotes, they might not have this problem because they sometimes have like circular DNA, and that's not a problem for them. And now I'll explain to you why that is important. Okay, so for eukaryotic chromosomes, okay, because we have linear chromosomes, each cell division is actually going to result in a bit of a shortening at the ends, and the reason for that is because of the whole RNA primer situation. Okay, so if you look at this diagram, right, you have the leading strand on the top, which is being created continuously, and this is the end of the chromosome now. Okay, so each of the RNA primer, so like you would have had a primer here and a primer here and a primer here. So all these primers are going to be replaced by a DNA polymerase that's coming up from the top, right? From the upstream, we say, right? So this red section here will be replaced by the DNA polymerase 1 coming from top here. Okay, so you're going to replace that with the blue stuff. But at the end of the chromosome, there's no DNA polymerase coming up from anywhere because this is the end of it already, right? And so there is no DNA polymerase 1 that will be able to replace this RNA primer. So this primer will just eventually fall off, and so you will have a dangling section here, and that part of the DNA is actually going to be cut off, and so the new DNA is going to be shorter than the original. Okay, so if this is allowed to happen, then each cell division will result in shorter and shorter chromosomes, and more importantly, it might start to delete important genetic information, right? And so to protect the ends of DNA, to prevent the erosion of genes, there are non-coding DNA sequences at the end called the telomeres. Okay, so telomeres are just repeats of 6 nucleotides. Okay, it's like TTA, GGG, and then it's just gonna repeat itself. So another TTA, GGG, TTA, GGG. It's gonna repeat like a couple hundred times, perhaps. And that telomeres kind of serve as the kind of like the plastic cap at the end of your shoelace, right? They prevent the DNA from fraying at the ends, and it delay the shortening process during DNA replication. Here in this picture, you can see the X in the middle are the chromosomes, and then the dots at the ends are the telomeres. Okay, so they protect the ends of your chromosome. So you might remember Dolly. Dolly was the first mammal that was cloned from an adult somatic cells using what's called nuclear transfer. So basically, you take the nucleus of a cell and you put it in an egg, and that's how you cloned Dolly. But it only lived 6 years, half of what was expected. So what they found was that the telomeres of Dolly was as short as that of the donor cell. Okay, so the donor cell was come from a much older sheep, and because that older cell has gone through many cell divisions already, so the telomeres in it was already quite short. So when you put it in an egg and you clone the sheep, even though the sheep was supposed to be born young, but on a cellular level, its DNA were already quite aged because of the short telomeres. Right, the cells has already gone through so many division in the donor cells, and so with critically short telomeres, Dolly was not able to live very long. So isn't that a common problem for everybody then? Right? If our cells are going to have shorter and shorter telomeres, does that mean every time we have a kid, right, when we reproduce and have a child, the child is going to inherit those short telomeres? Well, thankfully, that's not the case, because in germ cells, these are cells that are going to be dividing to become sperms and eggs. So in these germ cells, there is an enzyme called telomerase. Okay? And telomerase function is to re-lengthen the telomeres. Okay, so in a regular cell, the telomeres shown here in red would be shortened after each division, and when it gets to critically short, then the cell no longer divides. Okay, however, in germ cells, cells that will become sperm and eggs, they have the telomerase, so they kind of replenish the length of the telomeres after each division, right? So that way, when the sperm and the egg combines to form the baby will have fresh telomeres to start with, rather than inherit critically short telomeres. So that's why it's not a problem if you're not cloning with a nuclear transfer. But telomerase is actually linked to cancer. Okay, cancer in a very basic sense is basically uncontrolled cell division. Okay, the cell divide virtually indefinitely. And one reason that cancer cells can evade what we call cellular senescence and keeps on dividing is because they have high levels of telomerase. Remember, you're not supposed to have telomerase except for germ cells, right? And stem cells. But in cancers, for some reason, they're able to reactivate these telomerase, and so they are able to keep on dividing. In fact, if you look at cancer cells, the telomeres are much longer than that of regular cells. So researchers are trying to explore ways to inhibit those telomerase in the cancer cells as a potential cancer treatment. So that's it for DNA replication. In the next video, we're going to be looking at what happens after you replicate the DNA, which is for cell division, the actual process of mitosis. Thank you for watching.